All right, it is 2, so we'll get started. Earlier today, I should have put up your test grades on Blackboard. So what you'll notice is there's 2 columns. One is just like the raw score, and then there's another column that says test 1 score. That's your overall percentage, right? So that's what you want to focus on. You should be able to go back into your test now. It should be available, and you should be able to see what your answers were. And so we're not going to go over the test question by question right now. What I will do probably on Tuesday is I'll spend some portion of the time on Tuesday and make a little video overview of test 1, what the answers were, and all that. And then if you have any discrepancy on how I scored something, what I would ask for you at that point, after you see my review video on the test, just send me an email and let me know why you think you should have got more points on a particular question, and we can have that dialogue then. Yeah, so that's all there is really to say on test 1 without going into the actual like detail of the grades, or the detail of the problems rather. And so with that, like I said, look forward to that on Tuesday, but we're going to get right into it. We have quite a bit to cover today. If you remember all the way back on Wednesday, and if you're not sure what PowerPoint I'm on, it's the one that it's really old now. It's what we finished on Wednesday, and we looked at this slide where we were talking about residues. We were looking at protein sequences, lining up the same protein from different organisms, lining up their primary sequence and just seeing what amino acids are invariant, that is, they never mutate, which means they're super important. Which ones are conserved, that is, amino acids that can mutate, but they have the same characteristics, and which ones are hypervariable due to neutral drift. And hypervariable means that the actual amino acid in that position is not all that important. All that is important is that you have amino acid there. Good example is this line that I'm looking at where it's positive, negative, and polar. It's just a bunch of different amino acids. So it doesn't matter what goes there. So that's where we ended on Wednesday. And so we're going to get right into it. So hopefully by now, after test 1, we have a pretty good idea of our amino acids. So just to begin today, I have this very quick question. Now I can pull up a poll for it. So I want to know, if I was looking at valine, what amino acid do you think, or amino acids, I didn't make it multiple answers, so just select the amino acid you think would be a good substitution for valine. There is more than one answer. And B, what amino acids would be a good mutation for arginine? So we're looking for conservative mutations in both A and B. See if you can look at these amino acids, remember what your structures are, and determine, okay, what's good mutations in each case. And just as a reminder, even though not until the final exam will I ask you a question like this amino acid, what's the structure and what's the charge, it's still very important for us moving forward that we remember our amino acids. So do about 20 more seconds on this. So if you haven't figured it out yet, see if you can get conservative mutations for these amino acids. All right. So valine. Valine is what we call an aliphatic or non-polar amino acid. And I guess it would help if I plugged in my writing tablet so I could actually write. There we go. All right. So valine, it's aliphatic. It's nothing but carbons and hydrogens. Therefore, we are looking for a non-polar substitution to be conservative because valine's non-polar. So to be conservative, it has to mutate into a non-polar. Alanine is just a CH3 side chain. Leucine, a bunch of CH3s. Glutamic acid is negatively charged, so that's not a good substitution. That's not conservative. Isoleucine, a bunch of CH3s, so that's good. Asparagine, polar, so that's not a good substitution. So to be conservative for valine, which is nonpolar, you have to mutate it to a nonpolar amino acid. So alanine, leucine, and isoleucine would all be good choices. For arginine, arginine is positive. So the only conservative mutation for arginine would be lysine because they're both positive amino acids. All right. So I do have one more question that has to do with mutagenesis and talking about this concept of mutations in proteins and what that means evolutionary. And what you see when you look at experiments, site-directed mutagenesis experiments, what that means is that we can modify, or we do modify, the DNA sequence of a protein and mutate amino acids at specific sites. In site-directed mutagenesis, what we find is that you can often replace valine for glycine. So I can mutate valine to glycine, usually no problem, that's okay. The protein works and it's folded. However, I cannot go from glycine to valine. That won't work. The protein won't work anymore. Who can think of a possible explanation as to why that is the case? Why can I go to valine to glycine, but not glycine to valine? Anyone have any ideas? Valine has more carbons, H bonds, glycine is non-chiral. All right, so for hydrogen bonds, valine, the side chain of valine is CH3, CH3, and with a that. So valine can't hydrogen bond. Remember, if a hydrogen is connected to carbon, no hydrogen bonds. Glycine just has a hydrogen, so it doesn't have hydrogen bonds either. So they both can't hydrogen bond because they're both nonpolar. So that's actually okay. Chirality, yeah, glycine is achiral, but chirality in amino acids don't actually matter because everything is L. So it's not the achiral, but it has to do why it's achiral, and it has to do with the amount of carbons. So the side chain of valine is this big bulky carbon, 3 carbons together forming a V. The side chain of glycine is just a small little hydrogen. when we look at amino acids and proteins, they're very compact. If you look at the inside of a protein, there's not much empty space, if any at all, for the most part. It's just atoms are in close contact to each other. So if in my original sequence, I have a glycine, the side chain is just a hydrogen. That means all around that hydrogen, I probably have amino acids, like different elements right next to that hydrogen. And so if I try to replace this hydrogen with a big bulky group, it doesn't fit, right? The protein won't be able to adapt to that. But if it's the reverse, if I have, this big bulky group, and I'm trying to replace that with a little bitty hydrogen, well, glycine can fit in there, no problem. So it does have to do with the size of the side chain. Even though they might have the same characteristic, we also have to be aware that these amino acids have size. And so we can't, glycine to valine is usually not permitted in mutagenesis. So any questions about any of the ideas presented here or anything at all? Well, if you do have a question and you're still typing out, feel free to continue to do it and I'll answer it when I see it. Otherwise, I'll move on. And here is more of that idea to get us comfortable with substitutions. So on Wednesday, we looked at this big chart and we looked at the different positions and we're able to tell invariant, conservative, and hypervariable. So I want you to do that now. I have 10 positions. I want you to tell me what positions can't allow substitutions, which ones allow conservative, and which ones allow highly variable substitutions. Here I have a big long pole, so I can figure out, so you can tell me at every position, which category do you think it is. If the poll gets in your way, feel free to minimize it, answer it, and if you click the poll button again, it should pop it back up for you. But let's take a look at this. Based on my amino acids here, which positions are unable to tolerate substitutions, allow conservative substitutions, and are highly variable? A little bit more practice with our amino acids in one-letter codes. And if you have questions while doing this, as always, feel free to send me a message. You can always send me a private message. We'll do one more minute here before we go over these explanations. All right. So if you're still working, continue to work. I'm just going to go over the answers at this point. And a lot of the positions where we're feeling pretty good. So I'd say about half the positions we have a consensus, the other half, not really. So let's take a look at this then. There's my pen. Okay. So position 1, D and E. Well, D is negative, E is negative. So this would be conservative. I guess conservative is answer B. So that's conservative because it can be either negative. 2, so we got a couple of different answers here. I would say this is conservative. I and L are hydrophobic. Y and F are also hydrophobic, right? You could maybe make a little bit of an argument that these are what are called aliphatic. And that just means like carbons and hydrogens more or less in lines. And these are aromatic. And so there is something to be said about that. However, I think just by looking at the type of amino acids these are, this would be more of a conservative. Everything is nonpolar hydrophobic. And yes, even though Y does have a little OH on it, it's still hydrophobic. 3, this one is definitely highly variable. You got polar amino acids, you have a charged amino acid, you have non-polar amino acids. So we're all over the place here. So that would be highly variable. 4 is also highly variable. G's, E, A, T, it doesn't matter what's there. Anything can go there. 5, okay. 5 is an interesting case. When studying your amino acids, I told you that histidine is often one of the charged amino acids. However, it is only charged maybe like 30% of the time. And most of the other times it's just polar. So looking at this, I would say that is conservative because these are all polar amino acids. Usually when looking at this and you see a histidine, unless you actually know what chemistry it's in, well, even at that case, histidine, when it's in a reaction, and we'll look at this, it's the only amino acid that can act as like an acid and base easily. So whenever we look at histidine in a sequence, we usually just think of it as a polar amino acid rather than a charged one. So M and T are both polar as well. So I would say all of these are polar. So that would be a conservative. The easiest one are unable to tolerate because you just see something down the line. That means at position 6 for this protein, glycine is critical to function. Position 7, highly variable. You got some polars and some glycine. So it doesn't seem like to matter really what's in there. Position 8. That one you could say is, position 8 is probably the hardest one because it's all negative and then one like L for tuna. So It depends on how you want to debate that. You could say it's highly variable, but I don't know if I would be comfortable with that because you see so many E's. So it doesn't look to be highly variable. Maybe you can even make an argument for like A-ish and tuna is just an outlier. B, maybe conservative, even though L is non-polar, so it doesn't really fit that. So 8 really doesn't fit into a nice category, and that's what happens some of the times when you do this in real life, where you'll see same amino acid. One organism doesn't have that there, then same amino acid. So 8 doesn't have a clean answer, right? It depends on how you want to argue it, is what I would say for that. On a test situation, if I ask this question, I won't give you something like 8. I'll give you something like 9, where it's unable to tolerate that. Or I'll give you something like maybe 2, where it's like, oh, they're all nonpolar. So we have to see that. Or number 10, all nonpolar. So that's how I would list those. Any questions about our different substitutions here, looking at amino acids? All right, well, if you do have questions, continue to type them. Otherwise, I'm going to move on. And when we have a bunch of sequences from different proteins, we can look at the mutations from those proteins and make this phylogenetic tree. And we can see, based on how similar proteins are, we can start to say how similar species are. And that's what we're showing here. Here's a phylogenetic tree for the protein chromosome, cytochrome C rather. Cytochrome C found in all of these species. And we just look at their mutations, and based on how many mutations there are, we can see how closely the organisms are related. And so you can see by just looking at this one protein, we get a tree that makes a lot of sense, right? All the mammals are grouped together, birds are grouped together, fish grouped together, plants grouped together, fungi grouped together. And that's all based on a computer just looking at one protein. And these numbers there, it just says, based on 100 residues, how many mutations am I likely to see? So that's what all of these numbers are actually showing. And each of these node points is a common ancestor, right? So that's what the organisms split off. So that is just one way where we can use this information in a practical sense, looking at mutations. It's not only to see, how a protein functions, what amino acids are important for a protein. We can also look at how different organisms are related based on their protein sequences. And I did have a question when we make a tree. Skip. We need to make up some time. So we can just skip like looking at the sequence and making a tree. Just the more related you are, the closer you are on the branches. And looking at protein evolution in a whole, we can figure out some interesting things about proteins in general. One thing that we can learn about this is the existence of what are called domains. And domains, they're found in proteins, and they're a sequence that are conserved, like 40 to 200 residues. And these domains, they can have like structural similarities, even if the sequence is different between different organisms. So what's an example of a domain? So one example is called a zinc finger domain. And what a zinc finger is, and we'll learn about what I'm drawing right here, but I'm drawing what's called an alpha helix. A zinc finger is where you have an alpha helix on a protein, and you have this zinc metal that is interacting with the protein. And anytime you see a protein with this domain, you know that it will interact with DNA, right? So these domains are like little parts of the protein that do specialized functions. So zinc finger domain will interact with nucleotides, right? So that's what the idea of a domain is. And once you start looking at proteins, people talk about domains all the time. And when we're looking at different proteins, if we look at a certain domain, if the sequence is greater than 40%, so if they have more than 40% of the same amino acids, those domains probably do the same thing. However, if the domain doesn't have that grade of sequence similarity, they probably do different things. Another vocab that we need to learn is an ortholog. And a ortholog is a protein that has the same function in different species. So it's a homologous protein with the same function, but found in different species. If it's in the same species, that's usually due to a gene duplication. So one way that proteins can mutate is through these gene duplication events. So let's say, right, I have a gene for protein A, right? And let's just say there was some event that I duplicated my genome, and now I have 2 copies of protein A in my genome. So I have 2 DNA sequences for A. What that means is that this copy of A, for example, can mutate a lot. Like this can mutate all at once and the organism won't die, because as long as I still have a working copy of A, and if A is critical for the organism, I can mutate my other copy and still live. And what can happen after, a couple 100,000 to millions to hundreds of millions of years is that protein A now has a completely different function that gives the organism some kind of advantage. Now it's like protein B, and that organism has just evolved. So that's one way that gene duplication, this idea of gene duplication, can take homologous proteins in the same organism, mutate one copy of them, and now you have a protein with a whole new function. Now, this chart over here, what I'm showing you is I'm showing you 4 different proteins. Each one of these is a different protein. And these lines are, looking at common ancestors and how many years since you have diverged from that common ancestor. How many mutations per 100 sites can you expect? So we have some proteins that have wild variation. This says like, even after 100 million years, like 80% of the proteins have changed. While something like histone, if you look at 1 billion years since histone H4 has diverged from a common ancestor, you're looking at maybe like 1 amino acid change, 2 amino acid change. So that question of why, when looking at this, do histones change very little? Does anyone have like an idea of when we're looking at histone proteins, why might they not actually evolve over like 1 billion years? You want to have a hypothesis on that? It can't change because the residues are critical for its function. That is correct. But even something like hemoglobin, hemoglobin carries oxygen throughout the blood. And so it also has amino acids that cannot change. And histones interact with DNA. So why do histones in specific, why do they never change? So you are right, it's because residues are critical, but it shows like zero change whatsoever. Why does it show zero change, even though other proteins have critical things, but they also show some change? And I guess to really think about that question, you should ask yourself, what does a histone do again? What is the main purpose of a histone? Going back to our general biology. They do wrap DNA. So histones, correct, wrap DNA. DNA, as far as we know it, never changed. It's a very simple molecule, ATCG phosphates. So if your protein, if your whole job is to have a molecule wrap around you, and that molecule doesn't change, then you're not going to change either, right? Because, and it does go with like, well, that means every single residue in histone is critical for its function. Yeah, that's exactly what it says. And the reason we can logic that out is when thinking about, what's the function of a histone? It's a rock that you wrap DNA around. Like almost every residue in the histone interacts with DNA or interacts with other histones. So it makes sense this would not change. And then part B, I will go along and answer part B for us. Why do we have these domains? So the way I think about domains is that if you have a protein, a domain is a little area of function inside that protein. So like a protein can have like, for example, I'm drawing a protein with 3 different domains, and each domain has a different function. So that protein can do, 3 different things, more or less. So domain 1, domain 2, domain 3. They each have a different function. So what's the advantage of this? It's very easy to create new proteins from this domain system. It speeds up, more or less, speeds up evolution. Because if you think about it, during this gene duplication event, what can also happen is that maybe just a domain gets, the DNA of that gets duplicated. And now for some reason, and depending on how far we get this semester, we might get to this section, but sometimes your DNA just copies itself. It just copies domains, and it'll say, well, this domain is going to go, so domain 2 will now go in protein C. And protein C now has a new domain, and it might not work. It might kill the organism, or might give the organism some advantage. So it really speeds up the flexibility of an organism as a population when these domains are self-contained. They have self-contained function. So when making new proteins, I'm saying making in quotes because it's all an accident, but when duplication events happen, you can copy over these domains to new proteins. You might be able to gain a new function, and you can exploit a new niche, which means that your population will now outcompete the other population. So that's why domains exist. It's because the organisms that didn't have domains, if there were such organisms, they could not adapt as fast on a population scale. Does that explanation make sense to people, or are you confused at the stuff I just said, or any other questions in general? Right. Well, if you do have a question and you're typing it out, continue to do so. I believe that's the end of this PowerPoint. So I'm going to switch here. So let me get the other PowerPoint up. Second screen. That share screen. Yeah. So in the past, what feels like since the beginning of this class, but really like in the past month, we have been talking when in essence of proteins only about primary structure. And remember, there are 4 levels of protein structure. Primary is just the amino acid list of amino acids from N-terminus to C-terminus. We're going to talk about secondary structure now. Secondary structure are local areas of structure in proteins. We're mainly going to focus on what are called alpha helixes and beta sheets. These aren't the only secondary structures found in proteins, but they are the most common, and that's why we're going to focus on them. All right. So to talk about secondary structure, we have to go back to this idea of the peptide bond. All right. So what we're looking here is the peptide bond. So here is one amino acid. So I'm going to call that amino acid 1. This is a second amino acid. That's amino acid 2. Right? And you can tell they're amino acids because of the carbon alpha. Every time you see a carbon alpha, that means a different amino acid. And when you form a peptide bond, the peptide bond actually forms a plane. And it's like a square that I'm drawing here right now between the carbon alpha, carbonyl carbon of amino acid 1, nitrogen, and carbon alpha of amino acid 2. So this is amino acid 1. This is amino acid 1. This is amino acid 2. This nitrogen and that carbon alpha is amino acid 2. All right. And generally, this peptide bonds is in the trans conformation. And what I mean by trans is the side chain of each amino acid when you're following are in opposite direction. So amino acid, the side chain is pointing down. Amino acid 2, the side chain is pointing up. And the reason why this is because if the side chains were in the cis conformation, that is, they were both pointing in the same direction, you would get steric hindrance, right? The side chains could bump into each other. And that would be energetically unfavorable. Your exception for this are prolines. Prolines, 10% of the time, their bond is in the cis conformation. And that is because proline is special. And that's what this is. Why can proline be in cis? Well, it's because if you remember your proline structure, proline actually comes back and goes behind to bind on its own amine group. So if we had and what I'm drawing right here is a cis, this is a cis peptide bond the way I drew it because they're both pointing in the same direction. You now have space between these two because the side chain of proline is bending back on itself and binding its own backbone. So cysteine can be in these cis peptide bonds. Now, when talking about peptide bonds, we care about 2 what are called dihedral angles. And I'm going to erase all this. We care about what are called dihedral angles. and we call those phi and psi. So if you've never heard of a dihedral before, that is the angle between 4 atoms. Now, your classical angles are 3 atoms, right? A dihedral is 4. And we care about the phi and psi angles, right? And so phi is carbonyl carbon 1 to nitrogen 2, sorry. Here it is. Carbonyl carbon 1 to nitrogen 2 to carbon alpha 2. And these 2 just mean protein numbers. To carbonyl carbon 2. And for a dipeptide bond, or sorry, for a dihedral bond, anytime you see a dihedral, what you measure is the angle between the termini atoms, and it's the rotation between this bond. And the best way I can show this is if I actually leave the PowerPoint and go onto screen. So I'm going to do that. All right. So, and I'm going to get out my dual screen setup so I can actually watch myself do this. All right. So dihedral angle. You have 4 atoms connected. What you do is you take your 2 middle atoms and you line them up so you're looking down the middle atoms. So this is atom 2 right here and atom 3 right here, and you line them up. And then I'm going to say this is atom 1, and my other hand is atom 2. I know this is really hard to do on a screen, but bear with me. So with the 2 middle atoms lined up, you ask yourself, okay, what is the degree of separation between atom 1 and atom 4? Right now, I would say this dihedral has a measurement of 0 degrees. This is a measure of 90 degrees between my atom 1 and atom 4. So I would say this dihedral is currently measuring at 90. This is 45. If they're directly opposite, that's 180. This is 270, 360, or 0. So that's how we measure dihedral angles. It's made out of 4 atoms. Take the 2 middle atoms, you line them up and look down their center, and then you just see, okay, what is the distance, or what angle are atoms 1 and 4 making? That's how you measure it. So while I reset up the screen sharing here, does that make sense on what a dihedral angle is and how to measure it? Or did I lose somebody with that explanation? Okay. And you have another one, the psi, N2, Ca2, C2, N3. We're going to see, based on your phi and psi combination, based on what these values are, you get different secondary structure. So that's why it's super important. These angles will tell us, okay, what secondary structure are we seeing in this protein? And I should mention this image down here. The peptide bond between the C and the N, you cannot rotate, no rotation between the peptide bond, between the carbon and nitrogen. That's because this peptide bond, it has resonance. And so it has some double bond characteristics. It has like 30% double bond characteristic. So you cannot rotate around the peptide bond. And that's actually why we form this plane. And that's one of the reasons why we look at the angles that we do. Peptide bond itself cannot rotate. Right. So the first secondary structure we're going to look at, it's called the alpha helix. And before I get into that, let's take a look at the chart on the left. This is called a Ramachandran chart, named after the scientist who made it up, Ramachandran. What we're looking at here is on the x-axis, we have phi angles, our phi dihedrals. On the y-axis, we have our psi dihedrals. And what happens is that when you look at proteins in general, you'll see some correlation. And this is a very simplified Ramachandran. But a Ramachandran plot tells you, based on your combination of phi and psi, what secondary structure are you in? So if you have a phi and psi combination that is this, you know that part of the protein's in an alpha helix, and this is actually a right-handed alpha helix. Phi and psi combination over here, that's a left alpha helix. These are your beta sheets, and this C is for a different type of secondary structure we're not really going to get into. Like I said, there are more secondary structures than this. This is just a very watered down version. Anything in here that doesn't have color is usually not favorable. Blue is very favorable. And green's like, you can tolerate it. That's what the colors mean. And I actually did some work in this field myself, where I did different simulations of like glycine next to every single amino acid and just figure out what phi and psi angles do we see versus proline. You can see that in proline, where blue you cannot be. Proline has a lot less flexibility than glycine, which makes sense. Glycine is the most flexible amino acid because it just has a side chain of hydrogen. So you can adopt many different phi and psi combinations. Proline is bound to its own backbone, so very few side chain or phi psi combinations. Right, so that's what a Ramachandran is. Let's get to the alpha helix. And the way an alpha helix is, it's just a big helix. And the big thing I want you to remember for the alpha helix is that what holds it together and what holds all secondary structure together are backbone atom hydrogen bonds, right? So if we look at this alpha helix, here's a carbonyl carbon. Here's a nitrogen carbon alpha. And this is also a carbon alpha. So the carbonyl carbon of this amino acid is hydrogen bonding with the amine of a different amino acid. And every single amino acid has this, right? So if we look at this carbonyl carbon in the back, hydrogen bond with this nitrogen. This carbonyl carbon, hydrogen bonding with this nitrogen. So what an alpha helix is, it's a ribbon held together by backbone hydrogen bonds. And you hydrogen bond the N+4th residue. So if I'm residue 1, my N+4th is residue 5. So if I'm residue 1, I have a hydrogen bond with residue 5. If I'm residue 2, I'm with 6. 3 with 7, so on and so forth. One complete turn of the alpha helix, roughly 3.4 residues. So that's 360 degrees. The side chains of the alpha helix point down and away. They do not point to the inside. The inside is filled with backbone atoms. So side chains are down into the back. Only certain amino acids, certain amino acid combinations can be in alpha helixes. So this question, since we're running out of time, let's just talk about these briefly. So why is it unlikely that you would have like ARG, lyse, met, phi, trip, ti, val if you only had an alpha helix made out of, if you only had a protein made out of this, why are you unlikely to see an alpha helix? And so that has to do with the side chains, right? All of these amino acids I have listed have big, bulky side chains. And that means that if they are all pointing down, they're going to be bumping into each other. And you'll have steric hindrance. So you can't have side chains, nothing but giant side chains in alpha helix. You can have some of these amino acids, but you can't have them all because they will bump into each other, steric hindrance, unfavorable. So you need to have some small amino acids in there. Alanine is like the best amino acid at forming alpha helixes. For proline cannot be in the alpha helix at all, except the very first amino acid. So proline is called a secondary structure breaker because if we go back to proline, that's what proline looks like. It does not have an amide hydrogen. It loses the hydrogen on its backbone because the side chain is bound to itself. For secondary structure, one of the key features, like I said, is the hydrogen bond between the carbonyl carbon and amide hydrogen of different residues. So proline doesn't have this. It can't be in hydrogen bonds with its backbone, so it can't form alpha helix, and it can't form beta sheets either. So that's why you'll never see proline in a alpha helix. So if we're looking at sequences that are most likely to form alpha helixes, what we're looking for, one, sequences with no prolines. So here are just 3 amino acid sequences I have. And I'm just saying, okay, which one is most likely to form an alpha helix? It can't be 2. 2 has a proline in it. Prolines are secondary structure breakers. So now it's between 1 and 3. And neither one of these have a proline in it. So we just have to look at, okay, sizes now. So if we look at 1, we have a lot of positive charge right there. And we have some big bulky side chains. While in 3, we have some smaller amino acids, a lot of alanines. So when seeing, okay, what is going to make a good alpha helix, it's usually small amino acids, no proline, and also really no glycine. So glycine is also a structure breaker, secondary structure breaker, because glycine is so small, it has a lot of flexibility. And so it doesn't want to be constricted to being in an alpha helix. Once you're in an alpha helix, you can't really move. Glycine loves to turn. That's one of the features of glycine. It can move a lot. In a secondary structure, it can't. So glycine doesn't really like to be in that. So it would be 3. Can't be 1 with proline. Glycine, not really either. Big bulky things, and that's 3. All right. So there's your basics of the alpha helix. Again, the big key takeaways are: know that alpha helix is due to backbone hydrogen bonds. Know that the side chains point out and away. Know that in general, you want small amino acids in your alpha helix. You cannot have proline in it. In general, you can't have glycine. And you cannot have a lot of big, bulky side chains altogether. All right, so any questions about what we covered today? All right, so if you do have something, continue to type it out. Otherwise, that's all I have. Went over a little bit, so I apologize for that. Like I said, I'll be putting up like that test video probably on Tuesday, going over it, going over the answers. I'll be putting up a homework today for what we went over in class. Otherwise, hope you have a good rest of your Monday, and hopefully I'll see you on Wednesday. Take care, everybody.